Today I'm going to talk to you about a technique called staining. Staining is a technique used in biology and medicine that highlights cellular features and substances. This technique involves tissues and cells that are first fixed, and then they are adhered to a slide, and then they are submerged in solutions in one or more stains. So right here next to me is a automatic stainer. So in this there are little baskets containing a solution or a dye. There's a basket of slides that you can insert in here, and it travels through each dye or solution, and it depends on how long it's in each solution depends on what solution it is. So the solutions are used to take the water molecules out of the cells and also help the binding process of the stains, and then also it removes the excess stain from the cells. So for example, right here we have xylene as one of our fixatives of the stain, or alcohol removes the water. So, and then there's several different kinds of stains in this machine. That's a particular staining system. So what is stained, you might ask? Well, any cell or tissue can be stained, as well as any inorganic, or some inorganic components, I should say. So, for example, some cells or tissues that are stained are nerve cells, muscle tissues, fungus, cartilage, bone, you name it. And then some inorganic components that can be stained are calcium and iron. Iron specifically has a stain for itself only, but calcium can be bound to the stain or dye in routine staining, and it appears dark on the slide through the microscope. Okay, so where is staining used, you might ask? Well, in diagnostic pathology, they use this technique every single day to help identify infectious diseases, neoplastic diseases, autoimmune diseases, inflammatory response, etc. A pathologist will look at the organelles in the cells and determine what they are based on their structures, and also what's present, what's not present, that. Okay, and then also it is used in research, and researchers are looking at different cells and reagents that they react to and study that. So how it works. There are many different types of staining systems. So let's come over here and look at a couple of stains that I have out. We have eosin and hematoxylin. Excuse me. So these two work together, and they're a staining system. They are very common in histology. Hematoxylin is an acidic, actually it's a basic dye that binds to acidic structures in the cell. So hematoxylin will make that structure have a purplish blue appearance. So for example, the nucleus is an acidic structure in the cell, and this dye will turn that structure purplish blue. Eosin, on the other hand, is acidic, an acidic stain, and it attacks the basic structures, and it turns them pink or red. So in squamous cells, eosin turns their cytoplasm, because it's basic, pink or red. So some benefits from this technique, staining. It helps differentiate the different types of organelles. Of course, that's very important. Without staining, we wouldn't even be able to view the cells or their organelles at all. It also helps tell us about the viability of the tissue based on what organelles or structural details are there or not there. Some challenges with staining are that we have background staining. Whenever there's a high level, it obscures the organism or structural detail of that the observer is observing, and that's not a good deal. there's variability of fixation in the tissue sometimes, and that causes the stain to variate, and sometimes detail is lost whenever there's a variability in the fixation. Some reagents are pH sensitive, so it's kind of difficult to work with them whenever sometimes it's hard to control the pH of the cell. Then also a challenge is over or under staining. It causes a diagnostic problem if like a cell or structure or the whole tissue is overstained or understained. It can cause a false positive or false negative appearance. So whenever a pathologist is looking at their slides, they are looking for a positive stain, or a stain for a positive, and if it's overstained, it might look positive when it's really negative. So that's kind of how that's a challenge. But they have controls that are slides used to monitor the over or under staining. They place those in the basket with the slides being stained, and they stain them with them to, I guess, determine if the stain took correctly. So I told you about staining, and I hope that you learned a little bit about this technique used in biology.